Today, I'm glad to have our first speaker, Dr. Nobuto Takeuchi, from the University of Auckland. I give you the time. Hey, yes, thank you. Please start.
Thank you, Asian. Thank you for inviting me to this seminar. Really pleased to share our results and research with you guys. So today is my talk is about the evolutionary biology of so-called reproductive division of labor, which I'll momentarily explain, and that is driven by cheetahs, which I will also explain soon. And so reproductive division of labor refers to a biological phenomenon, biological situation where individuals or replicating entities divide the labor between reproduction and helping reproducers reproduce. So these individuals are assumed to form a group, and so we have individuals forming a group, and so each dot would be individuals, and so a certain fraction, typically a very small fraction of individuals, serve as reproducers, which then replicate and transmit genetic information across generations over different groups. So when groups split, reproducers reproduce. And then the rest of the individuals are called helpers that assist reproducers to transmit genetic information, but themselves are not capable of transmitting genetic information across multiple generations. Even though they may replicate momentarily, their lineage is a dead end. And examples will illustrate more precisely what this phenomena is like. And most probably the famous example of the reproductive division of labor occurs at the cellular level within multicellular organisms. So the single multicellular organism consists of a population of cells, and there are two types of cells: germline cells that transmit genetic information, so they are reproducers, and somatic cells that assist germ cells to transmit genetic information by performing various somatic tasks. So it is illustrated here schematically. So you have this germ gametes unite and form a zygote, and then soon the population of cells, as they divide, split into two groups. One group is germline cells, which will generate next generation, and then the rest will be forming somatic cells that do the job to help reproducers reproduce, but themselves do not transmit genetic information across the generations of multicellular organisms, even though they may replicate temporarily within a group. Another famous example is called eusociality, or exemplified by ants, termites, bees, and so forth. And so eusocial insects, they form a colony. And so colony consists of many individuals of insects, and there are two types of insects. One is queens that transmit genetic information by laying eggs. And so this is a queen. And the queen lay eggs, and then most of the eggs will develop into so-called worker individuals, worker bees, worker ants, that assist queens in laying eggs and reproduce. For example, by defending a nest or taking care of brood, taking care of their siblings, and collecting food and so forth. And another example, just the final example I want to mention, even though there are more, is somewhat unusual. It is a reproductive division of labor at molecular level within a single cell. So the cell is a population of molecules. You could conceive cell as such. And there are two types of molecules, broadly speaking. One type is a genetic information transmitter, so genes, genetic molecules, and all the rest are either directly derived from genes or produced by the enzymes. So these other molecules assist genes to transmit information. But the enzymes themselves, even though when the cell splits, enzymes you might think are inherited, but over a long period of time, over many generations of cells, the enzymes will be diluted out because enzymes don't replicate. So enzymes, even though they might be transmitted temporarily, they do not transmit genetic information. So in this sense, you could say central dogma of molecular biology is actually one specific example of reproductive division of labor. And so reproductive division of labor has evolved at vastly different biological scales. So starting with molecules like angstrom to micrometer to centimeter. So it's at very different scales. The same thing happened again and again independently. So the natural question is, like, is there anything driving this across different levels of biological hierarchy, something that is common across different hierarchy? And so they're thinking about how reproductive division of labor comes about. As an evolutionary biologist, we encounter a certain apparent paradox. And that is that according to the logic of natural selection, as explained by Darwin, individuals we expect maximize the self-reproduction. So individuals should maximize the fitness. But the reproductive division labor happens in a system where individuals perform so-called altruism. So the altruism is a social behavior action taken by individuals that decreases the fitness of the performer and increasing the fitness of the recipient. So, for example, somatic cells, they won't reproduce, but then instead they help germline cells reproduce. So that will be altruism. And a system like that, where individuals cooperate with each other, even to the extent that they sacrifice their personal fitness, these cooperative systems are vulnerable, susceptible to invasion by cheating individuals, the individuals that avoid cooperation, avoid altruism, and instead uncontrollably replicate within the group, which will eventually decrease the fitness of group, and the group will collapse. So even if reproductive division whatever might be a good thing for group at group level performance might increase, but then cheaters can invade and destroy groups. So how is it possible that in the light of the logic of natural selection, how is it possible that reproductive division of labor evolves? And the classic idea to explain this is called kin selection theory, which is proposed many years ago, more than half century ago, centuries ago. And this kin selection theory posits that there is a parameter of describing structure of population called relatedness that can explain this phenomena. So this relatedness refers to how individuals within a group are genetically similar to each other relative to individuals between groups. So imagine individuals are forming groups. So we have these 4 groups here. So each dots are individuals. And different colors, let's say, indicate different genomes, different genotypes. And if this genetic variation is partitioned in such a way that all the differences are between groups, but within group there is no differences. So the variance between groups is high, but variance within group is small. So variance can be decomposed into between group variance and within group variance, and if the within group variance is small, but between group variance is high. So that's the situation where individuals within groups are highly related. And in this case, natural selection can favor altruism because then helpers will, the individuals that perform altruism will interact with each other, and cheaters are interacting with cheaters within the same group, and then group containing more altruistic individuals will perform better than the group containing only selfish individuals. So this way, natural selection can select for altruism, and vice versa. So if genetic variation is more seen within group than between groups, so between groups there is no difference between groups. They have a similar genetic constituency, but within group there is genetic variation. In that case, selection will favor cheating, so that the reproductive division of labor will not evolve. So the classic theory therefore posits that the high relatedness, so situation depicted here, is a necessary condition for reproductive division of labor. So that's the classic theory. And in this talk, I want to present our research a model, in fact, a theoretical model that predicts the total opposite of this classic theory, and namely, the reproductive division labor evolves when relatedness is sufficiently low. Or more specifically, the model demonstrating a pathway, a novel pathway to the evolution of our reproductive division of labor, but that pathway is possible only when relatedness is sufficiently low. So this is the conclusion of my talk, and I'm going to explain how we came up with this conclusion. So let me explain in a single slide our theoretical model. It is originally individual-based computational model, and so the model is depicted here, and so model consists of a population of replicating, reproducing organisms. So we are thinking about some multicellular organisms, and each multicellular organism consists of a population of cells. So these guys are cells. You could also imagine these are primitive cells, and each cell containing replicating molecules inside. That's also okay. If you more like replication network, that might be something you want to think about. But either way, so and then when this replicating smaller individuals, when they replicate, then at some point the group will split when the number of replicating individuals reaches certain threshold, then the group will split. But just maybe for fixing terminology, I will talk about organisms and cells instead of replicating entities and groups. And then organism can also shrink when cells are not replicating well, and then they can also die. So there will be a competition in terms of which organisms can replicate faster and grow faster at the level of organisms. But also there is a competition within organisms, because these cells are also replicating. So those replicating faster will be able to leave descendants within the group as well. And so this is how cells replicate. So we think of two types of cells. And so the P and Q, imagine P and Q indicate different cell type. And so P can replicate, and it sometimes can also convert its cell type into Q, and Q can also convert back. And this parameter K, that indicates how much investment a cell makes to a somatic function. So we are assuming each cell is allocated a finite amount of resource to invest, and cell can either invest in performing a somatic function, that's increasing a fitness of everyone within the group, and then the remaining amount of resource. So the amount of resource is a sum to 1 when you sum up. So the remaining amount of resource is invested to self-replication. And initially, we assumed that P-type cell and Q-type cell are identical in terms of their resource allocation. So there is no functional difference between cell types. And our question is, when we let these cells evolve in terms of how much resource they want to allocate, and how fast they convert their cell type, and we want to know how this distribution of these parameters change. So since this is an individual-based model, we have every single cell represented in a model, and every single cell has its own unique resource allocation strategy and cell set conversion, but this can mutate when the cells replicate, so that's how the distribution can change. And here's a definition of the fitness, or perhaps it's better to say it's a definition of the rate at which cell divides, which is a multiplication of two factors. So one factor, the average, this is the how much all cells within the group, a single group, invest in somatic function. So more cells invest in somatic function, then the faster the average cell replication within that group. But the individual cell replication rate is also multiplied with the individual personal investment in self-replication. So that means that on average, cells do well when more cells invest in public in somatic function, but individual cells do better if they invest more in personal reproduction, because if all other cells invest in somatic function, but I, let's say, don't invest at all, maximize my self-replication, then I enjoy faster replication within that group than the other groups. And so that's why there is a selection, natural selection to minimize investment in somatic function within group, because that will make my replication faster. But if all of my fellow cells are not investing in somatic function, then average replication will drop. So my multicellular organism to which I belong to will not grow very well. And so then as a group, my group will be doing bad. So at the level of competition between organisms, it's good to invest more in somatic function, but personally, as a cell, I don't want to invest. So there is a conflict of depending on the levels, there is a conflict. So the question we want to ask using this model is like, does when reproductive division of evolves in this model, does it evolve at all? And so we tried out this computer simulation with different mutation rate and different so-called split threshold. So this is basically the number of threshold number of cells at which organism splits. So basically, this is a parameter defining population size of cells per group, which is important for how much genetic variation there will be within an organism, because if you have more cells, then more genetic variation. And we knew from separate work that these parameters are important for controlling the relatedness that I have introduced in early introduction. So that's why we are varying these parameters to see whether reproductive division of labor evolves when relatedness is high or low. So we vary that, and we see a kind of a sort of a phase transition here. So in this region of the parameter, so low mutation rate and low split threshold, and cells remain basically symmetric. So there is no differentiation between cell type P, cell type Q. And in fact, actually, organisms mostly consist of just one type, just purely P. And type Q is basically not really present. And because it's not present, then the parameters can undergo very wide fluctuation because they are not present. But as these parameters increase in either direction, then the symmetry breaking occurs. So initially, P and Q have identical characteristics in terms of how much they invest and how quickly they convert to each other. But then they evolve this asymmetry. So this is a schematic illustration of what's happening at the end of the simulation. By the way, this is a log plot, so it's a different time scale. And so so this situation is indicated here. And so I will just zoom in. So at the end of the simulation, the P cell type, so when cell is in state P, then the cell does not invest at all in somatic investment. Whereas when P converts into Q, and then a cell invests in somatic function, and once a cell differentiates into Q, then it never reverts back. So the differentiation is irreversible, even though initially the conversion is symmetric. So that means that this is a germline cells, or this is somatic cells, or this is information transmitters, and this is information utilizers. So this is the reproductive division of labor evolving in this region. And then we yes,
can
please.
I ask a quick question?
Yes, of course, yes.
So the mutation rates, are they still symmetric or
Yeah,
not?
it is symmetric in the sense of all parameters indicated here. So we have basically 4 parameters per individual, and they all mutate in the same way. So in that sense, it's symmetric.
So
But it is not
DP is equal to DQ. That is right.
DP and DQ mutates in the same way, but the mutation is independent. So they are independent random variable multiplied to these parameters when they mutate. So they are undergoing independent random walk in log scale. So because it's a log scale random walk, so it's kind of biased towards 0. So there is a maximum parameter that they cannot exceed. So there's a reflecting boundary at the maximum parameter and to 0. But it's a logarithmic scale, so it's kind of asymmetric towards 0. In that sense, it's asymmetric, but every parameter is independent of each other when they replicate. And the initial condition is all parameters same.
Thank you.
Yeah.
I also have a clarifying question. How do we link relatedness in this model to mutation rate and the threshold?
Yeah. So we haven't talked about that yet. And so the relatedness is defined as the variance between individuals. How much of the variance you observe between groups and how much of variance you observe within groups. And this ratio, fraction actually, it's actually fraction, and that's the relatedness. And so that when you increase the group size, so when you increase the threshold at which organism splits, then population size of group will increase. And that means you have more variation within group. So that will influence relatedness. And if you change mutation rate, that also influences. And what we did is we are not setting relatedness as a parameter. We set mutation rate and split threshold as parameter, and we basically measure relatedness using. I can explain this more details, but using a sort of standard technique of measuring relatedness by using some random variable and regression coefficient, so forth. Yeah.
Thank you. Let's clear.
Okay. All right. So that's what we did. So it's very good that you asked this question. So that's what we did. And we measure relatedness over time because relatedness turns out changes over time. So at time 0, relatedness starts at low value. So more variation within group than between groups. But as reproductive division of labor evolves, the relatedness increases. That means that there is less and less genetic variation within group, but more and more between groups. So there is more genetic homogeneity, so to speak, within the group. And at the same time, this is the average. So this line is the average cell division rate. So that also increases. It's correlated. So basically meaning that the average fitness of entire system increases as reproductive division of labor evolves. And we don't observe this kind of change when reproductive division of labor does not evolve. And you can see the relatedness here is much higher than here. So relatedness, once reproductive division of labor evolves, relatedness is high after reproductive division of labor, but before that, it's actually lower. So we then tested, like, do reproductive division labor generally evolve when relatedness before reproductive division of labor is high or low? Because reproductive division of labor evolves, that changes relatedness. But we want to know whether reproductive division labor evolves when relatedness is low or high. So we have to think about relatedness before reproductive division of labor. It's sort of, because the evolution of reproductive division of labor itself changes relatedness. So we to investigate whether relatedness should be high or low for reproductive division of labor, we have to measure relatedness before something happens. So then it turns out that this is relatedness when reproductive division of labor doesn't evolve. That's that. And then this is the relatedness. This is before reproductive division of labor, and this is after reproductive division of labor. So in all these cases where reproductive division of labor evolves, the relatedness has to be low for reproductive division of labor to evolve. But once reproductive division of labor evolves, then and the relatedness is quite high. And so this means that the classic theory and our prediction are compatible. Okay, so high relatedness and reproductive division of labor coincide. That's the classic theory actually. But the interpretation of the theory was that first you have to have high relatedness, and then reproductive division of labor follow. That was the interpretation of the theory. But the theory only says, well, you have two things have to coincide. Our model says, yes, it will coincide, they will coincide, but the order is different. The first, reproductive division level is not there, then relatedness is low, and then reproductive division level evolves, and then relatedness increases, and therefore high relatedness and reproductive division level coincide. So it's compatible with the classic theory, but the causal relationship is reversed. And then, so why is it that the reproductive division of labor evolves when relatedness is low? Or why is it that the reproductive division of labor increases relatedness? Why does it decrease, within group variation and increase between group variation? So we came up with the following hypothesis. So when reproductive division of labor is not occurring, so RDL minus case, then all cells are basically reproducers. There is no distinction between reproducers and helpers, so therefore all cells are reproducers, meaning that every cell has a potential to leave descendants in the future, meaning that every cell has a potential to evolve a lineage of cheetahs within that group, because they all have a potential to leave lineage indefinitely. Whereas when reproductive division of labor evolves, then only a small subset of cells can leave lineage over many generations, which basically means effectively the population size is smaller. So only individuals that matter for evolution are those that will leave descendants in the long run, so then population size will shrink. So effectively, there is a kind of bottleneck going on. So only everyone derives from a small number of reproducers, and therefore there's a bottleneck effect, even though the group size is sort of more or less stay constant as they grow, divide, and grow, divide, but constantly, all individuals are, descending from a small population, and therefore there is a constant bottleneck going on, and that will then effectively decrease population size, and therefore makes the population more homogeneous within the group, and that's why relatedness increases. Okay, so that's, and so then we thought, okay, so that would mean that the hypothesis, so this is our hypothesis, that what if reproductive division of labor evolves, then that increases relatedness, meaning that if the relatedness is low, then there is a benefit in increasing relatedness, and therefore reproductive division of labor evolves. But if relatedness starts high, very high, then there is no point in evolving reproductive division of labor in this model. So that's why reproductive division of labor doesn't evolve when relatedness is too high to start with. So that was the hypothesis. And the prediction of our hypothesis is that, well, definitely, so if the reproductive division labor evolves, then the cheetahs cannot, well, they might evolve, but then the evolution might be slower. So that means that the average fitness of organisms across many generations will stay high if reproductive division of labor is present. But if the reproductive division level is absent, then the fitness over a long time will decline. So we did some kind of experiment, a computer simulation test to test this hypothesis, and I don't think I have time to explain this. I will skip. So yeah, I guess I will skip that. So yeah, so that was all I need to say. So basically, the reproductive division of labor evolves as a mechanism to protect cheetahs. Okay, it's a mechanism to increase relatedness, so that will make it harder for cheetahs to evolve because population size is smaller, less genetic variation, and so that's the advantage of reproductive division of labor. And I think I'll stop here because it's just 30 minutes. And acknowledgement, my collaborators, and one of my PhD who is now in Tokyo, Dominic, and my collaborator, Austin Gandhi, and also funding. Thank you. Sorry for rushing at the end.